In this lecture, we're going to talk about bacterial reproduction and how microbiologists measure the growth of microbial cultures. Before we do that, though, I'd like to take a minute to connect this lecture with our metabolism unit by talking about just how important all of the catabolic machinery of the cell is in order to allow something like reproduction, which requires a ton of anabolic processes in order to make twice as much of everything inside of the cell, so that when reproduction occurs and the daughter cells are formed, each of those cells has all of the things inside it that it will need to survive and thrive. So basically, the ATP requirements of the cell that we discuss in the metabolism unit are necessary for growth and reproduction. So growth, in the case of microorganisms, we're going to find in this chapter, is not growing larger so much as it is growing and dividing and creating offspring in a process that typically involves the cell dividing into two genetically identical daughter cells. And because they divide into two and they split, that process is called binary fission. In addition, prior to that binary fission, the manufacture of all of the cellular components that are required for each of those cells is made, and that utilizes ATP. And when binary fission or asexual reproduction isn't occurring, the cell is using ATP to repair cellular components because of that process of entropy, which you talk about in general biology, where everything sort of tends toward chaos. There's a constant input of energy needed to keep that cell functioning well. So I want to just take a moment to relate what has to happen in order for binary fission to occur and talk about how much that costs in terms of ATP. So first of all, when you're undergoing binary fission, you need to replicate the chromosome. So in a typical bacterial cell, the chromosome is about 3 million nucleotides. Notice here that in order to replicate DNA for every base pair you add of those 3 million, there is a requirement for 10 ATP, which means to replicate the chromosome takes about 30 million ATP. So no small amount of catabolism is required in order to just make the DNA. In addition, the cell needs to make all of the proteins that will both become the structure of that new cell. Think of something like peptidoglycan, and all of the enzymes that cell is going to need in order to do all of the jobs that the cell needs to do. And in order to transcribe each of those proteins, you have an ATP requirement of about 5 ATP per nucleotide. Translation costs about 3 ATP or more per amino acid that's added to the growing polypeptide. And if we look specifically at cell wall synthesis, which is going to be a big part of binary fission because the cell elongates to about twice its size and then divides, it takes about 4 ATP per sugar portion of that peptidoglycan. So you have the requirement for transcription and translation of each of those proteins, and you have 4 ATP required for each of the sugars. So it's energetically an incredibly expensive process to undergo binary fission, but it's the only way to get your genetic information passed on to the next generation. And so it's a cost that bacteria are willing to shoulder. Now, though there are a couple of different ways that prokaryotes replicate, we're going to talk specifically about something called binary fission, which I mentioned earlier, which is where the cell undergoes this process of elongation, replication of the cellular components, and then division into two completely distinct cells. Now, this, like many things, is something that is easier to understand when it's happening dynamically, when you're watching the process occur. So I'd like you to take a moment to watch how this process occurs and what it looks like in the cell.
Most bacteria reproduce by a process called binary fission. During binary fission, the parental cell divides to form two daughter cells. In the first step of binary fission, the bacterium replicates or copies its chromosome and attaches each identical copy to a separate location on the cell membrane. Unlike eukaryotic cells, bacteria do not need to dissolve a nuclear membrane or assemble a mitotic spindle, making binary fission very quick and efficient. Second, the cell elongates. Since the chromosomes are attached to different locations on the cell membrane, they separate from one another. Third, the bacterium pinches off at the center and forms a septum, a separating wall between the two compartments of the cell. Finally, the cell splits into two new daughter cells. These daughter cells are fully mature bacteria that can grow and, if conditions are right, continue the binary fission process.
Now that you understand how binary fission occurs, I want to talk about something called biotic potential. And biotic potential is not something that's specific to microorganisms. Any organism that's capable of reproduction has a biotic potential, the potential to grow and populate a given area. The biotic potential of any organism is dependent on two major things. The first is how many offspring you have with each reproductive event, and the second is how quickly you're able to make that reproductive event happen. So in the case of binary fission, the number of offspring is always doubled from the parent number. So one cell becomes two cells, and there's no changing of that. There's no multiple offspring. However, microorganisms, bacteria and archaea that undergo binary fission can have an incredible biotic potential because they are able to reproduce so quickly. They are able to move from one binary fission event to the other very, very quickly. And so their biotic potential has nothing to do with the number of offspring they have per reproductive event. It has everything to do with how quickly they can move from one reproductive event, go through all of the rigmarole of making all of the pieces that are necessary, and then undergoing another binary fission event. And just as a point of reference, Clostridium perfringens, which we're going to see in a few moments, is a wound pathogen, can undergo binary fission every 6 minutes, which is crazy when you think about the amount of energy that needs to be stockpiled and used to make all of those pieces, and then to actually go through the process of binary fission. That can happen every 6 minutes means that Clostridium perfringens has insane biotic potential. Now, to look at something that's a little more routine, something like E. coli has a generation time of about 20 minutes. Now, that may seem like a lot less compared to Clostridium perfringens, but if an organism is capable of reproducing every 20 minutes, that means every hour they can undergo 3 rounds of binary fission, which means they can undergo 20 rounds of binary fission in less than 7 hours. And what that means is that cell can grow and divide and become over 1 million cells in that 7 hours. So one cell can become 1 million in less than 7 hours. And that, my friends, is a huge amount of biotic potential. So the biotic potential of prokaryotes is not due to the fact that they can have multiple offspring during a reproduction event. The biotic potential of microorganisms is due to the fact that they undergo these binary fission reproductive events so very quickly. And that's not true for all microorganisms. Something like Mycobacterium tuberculosis takes about 15 to 20 hours to replicate, and there are some environmental bacteria who take 60, 80 hours in order to replicate. So there are some very slow growing microorganisms, and we would simply say that they lack the biotic potential of those that can undergo binary fission more quickly. Regardless, we attribute the biotic potential of microorganisms to their rapid generation time. When microorganisms are introduced into a new environment, they follow the same kind of population growth curve that any other natural population exhibits. So what you're looking at in front of you is a population growth curve that should look familiar because you typically talk about this in the ecology portion of general biology. And population growth curves are exemplified by a population moving through 4 predictable phases. They are the lag phase, the exponential or log phase, the stationary phase, and the death or decline phase. And always these population growth curves are plotted with the number of cells on the y-axis and time on the x-axis. Notice time has no units, the number of cells has no units. So what we're doing is just talking relatively about the number of cells over time and whether it's increasing or decreasing. Now, anytime you have a straight line that moves across parallel to the x-axis, you would say that that line has a slope of 0 and there's no change in the y value for a change in x, which means, right, which is a fancy way of saying that as time moves on, during the lag phase, there's no increase in the number of microbes registered or counted during this procedure. Now, what I want you to understand is that though there is no binary fission going on, there's actually a ton of activity. Those cells are taking in nutrients, they're undergoing glycolysis and the Krebs cycle and the electron transport chain, right? Or maybe they're just growing fermentatively. Whatever they're doing, they are undergoing all of those catabolic processes that they are capable of in order to make the ATP that they need to make their DNA, to make their RNA, to make their proteins, to make their peptidoglycan, to make all of those pieces, and that takes time. So the lag phase is sort of a misnomer. Nothing's lagging. There's actually a ton of activity going on here. What we're viewing here, though, is just the fact that all of that activity at this point does not result in binary fission. However, when the cells get to a point where they are capable of undergoing binary fission because they have all the energy, they have all the pieces, now you see a slope that is positive, meaning that as time goes on, increasing numbers of cells are being recorded. So that positive slope says to us that for a given unit of time, we have an increase in the number of cells. And in fact, that increase is exponential. Remember from math classes, exponential means a doubling, right? So by definition, if you undergo binary fission, you are doubling the number of microorganisms for every reproductive event. So we call this the exponential phase. A lot of textbooks refer to this as the log phase because if you plot the growth of the bacteria over time using the log of the number of cells, as is done here, you will have a slope of 1. So they call that the log phase, but what you want really to be careful of is that you don't think log and forget that during this phase, they are growing exponentially. So there's a tremendous amount of activity, and that activity is primarily binary fission. And that continues to happen so long as space and nutrient availability and all of the good stuff the bacteria need is there in abundance. They grow and grow and grow and grow maximally, but of course, just like any other natural population, they get to a point where they reach what's called the carrying capacity of the environment, the point at which the environment can sustain no further increase in the population number. So as they move from the exponential phase into the third phase of growth, you see again a slope of the line that's parallel with the x-axis. So there's no increase in the number of microorganisms, no increase overall in the population. Now, that doesn't mean that binary fission has ceased to happen, that means is for every cell that's undergoing binary fission, there is a cell that is dying. And so the level of binary fission is just enough to replace those dying cells in the population. So a very different thing is happening here during the stationary phase than was happening during the lag phase. During lag, lots and lots of activity. During the stationary phase, not very much activity, just enough binary fission to keep that population number at a even level, right? Because remember, when we're looking at this, we're not talking about the growth of an individual cell. We're talking about the growth of the population. So during lag phase, no increase in the population number. During the exponential phase, an exponential increase in the population number until things start to get not so good, and then the cells level out in the stationary phase, where as cells are undergoing binary fission, equal numbers of cells are undergoing death. Now, that will proceed for a while, but ultimately, the nutrients will become so scarce, the space will become so scarce, the toxic byproducts of metabolism will build up and crowding will occur, and ultimately you will get to the point where more cells are dying than are undergoing binary fission, and so you enter what's called the death or decline phase. And that's a natural phenomenon that happens in a closed environment. We're going to be talking at times during the semester about trying to manipulate this natural population growth curve, but for right now, all we want to do is talk about the fact that microbial cells, when they're introduced into a new environment, go through these four predictable phases: lag phase, exponential phase, stationary phase, and death phase. As they move through there, you want to be thinking about what is happening to cause either the increase, the decrease, or the lack of change in the number of microbial cells over time. So what we're going to be dedicating a lot of our time to for this lecture is how we count the number of microbial cells, right? Because it's not like a macroscopic organism where you could just simply point your finger and count each one that you encounter. You have to have a way of seeing these microorganisms and determining whether or not the population is increasing over time. And we're going to be discussing a couple of methods that we differentiate into two categories. The first category is whether that test is direct or indirect. And actually, we're going to look at a second category. We're going to look at whether that test can tell us the number of viable cells which are capable of growing and undergoing population growth, or whether it's a total count, which would include both viable and non-viable cells. So we're going to look at a couple of different methods. And what I want you to do is look at each of those methods and understand the basics of how that method is used to count microorganisms, whether or not that method is direct or indirect, and whether or not that method is viable or total in its count. The first of those techniques that we're going to talk about is a direct viable method, and that is something called serial dilution and spread plating. Serial dilution and spread plating allows you to see viable cells because you put those inoculations onto media and allow them to grow into colonies. You know that what landed on that plate is a colony forming unit, which is a viable cell capable of reproduction. Now, the problem, if you have a bacterial culture growing, is that typically the number of microorganisms in that sample is going to be so packed that your entire plate would look like the primary zone of your streak plates, right? Think about your streak plates that you were evaluating in lab 4. Those streak plates take in one single loop full of microorganisms, which is typically around 10 microliters of fluid from the sample. And when you smear that over the primary zone of a plate, you get so many bacteria there that you can't distinguish individual colonies. and there's so many that you can't count them. So what you need to do is dilute the culture prior to plating it so that you can dilute it to the point where individual colonies are visible and countable. So whereas in the streak plate technique, you use your sterile loop to spread out and dilute the bacteria on the plate. In this method of serial dilution and spread plating, we don't dilute on the plate, we dilute in liquid media and then put each of those dilutions onto a plate to look at it. And typically, this occurs by first withdrawing 100 microliters of sample from whatever culture is growing. And you take 100 microliters out because you don't want to take so much out of the sample that you cause a change to what's going on. So you want to take as small a sample as possible. So you take 100 microliters out and you place that 100 microliters into 9.9 milliliters of water. Now, in order to understand this math, you want to remember that 100 microliters is equivalent to 0.1 milliliters. So when I take 9.9 milliliters and I add to that 0.1 milliliters, my total volume is 10 milliliters. So I have a dilution of 0.1 mil to 10 mils, which is the equivalent of 1 to 100 in relationship to what you have in your actual sample. So what's in that test tube is 1/100th of what's in the sample. Now you'll remember from the very first lab we did that 1/100th as a fraction is equivalent to 10 to the minus 2 in scientific notation. So typically, we don't say this is a 1 to 100th dilution. We say this is a 10 to the minus 2 dilution. What's in this first tube is diluted 10 to the minus 2 as compared to the culture. Now, typically, that will still be too concentrated for you to see individual colonies, so you will undergo a series of dilutions. And the thing that is very important here is that the dilutions occur not going back to the original sample, but are dilutions of just the previous tube, right? So think about this just like that streak plate. A couple of the errors that happened in the streak plate had to do with going back and getting organism again, or not sterilizing the loop, which meant there were more organisms on the loop than should be there. And what happens when you do that is that you end up with colonies that are too close together. So notice, just like when you do your streak plate technique, and you streak the second zone only going into the first zone, and then you streak the third zone only going into the second zone, in order to make this second tube, you're withdrawing a sample not from the undiluted sample, but from that first dilution. So here we take out 1,000 microliters from this first tube and place it into 9 mils of water. Again, in order to understand how this dilution works, we want to remember that 1,000 microliters is the equivalent of 1 mil. So now we have something different. We have 1 mil in 10 mils total. We have a 1 to 10 dilution. So initially we had a 1 to 100. Now we have a 1 to 10. But we have to remember, and we have to signify here, that what's in that second tube is not a 1 to 10 dilution of the actual sample. It's the product of both of the dilutions. So it is a 1 to 100 and a 1 to 10, or a 1 to 1,000, which we express in scientific notation as 10 to the minus 3. So this, though it is by itself a 10 to the minus 1 or a 10th dilution, cumulatively at this point we have a 10 to the minus 3 dilution, right? So we in the first tube have a 1 to 100, in the second tube we have a 1 to 10, and together those give us a 1 to 1,000, which is 10 to the minus 3. If we plate that again, there's going to be so many colonies there that they're going to be too many to count, so we further dilute, taking from the second tube into the third tube and creating another 1 to 10 dilution. Again, that's cumulative, so we end up with a total dilution of 10 to the minus 4, which gives us a decent number of colonies, but we don't know that that's going to be the case, right? So we do we do another 10 to the minus 1 dilution and end up with a plate that's countable, and then we do another 10 to the minus 1 dilution in order to get a plate that's countable. So when we do this, we end up with these series of dilution. The first tube is 10 to the minus 2, then minus 3, minus 4, minus 5, and minus 6. And then we plate 100 microliter samples. And that's not to confuse you. That's because 100 microliters will spread and soak into the surface of the agar much more quickly than a mil. But because we're only plating 1/10 of a mil, we have to accommodate for that. And so these plates, though they come from tubes that are 10 to the minus 3, minus 4, minus 5, and minus 6, the plates are actually more dilute. So that first plate is diluted 10 to the minus 4. The second plate is diluted 10 to the minus 5. The third plate is diluted 10 to the minus 6. And the fourth plate is diluted 10 to the minus 7. When you put those plates into the incubator, you will look at all of the media that you've used to do that and recognize one of the drawbacks of this method, which is it's incredibly time and media expensive, because all of those tubes with the green caps there are all just going to be discarded. The only thing that's going to be kept is those 4 plates. So you put those 4 plates into the incubator and you have to wait for them to grow, which gives you the second drawback of this procedure, and that is that you have to wait for the results. When you get those results 48 hours later, you end up with 3 plates that are fairly useless to you because they either have too many colonies to count or statistically too few colonies to count. Too many colonies to count would be 300 or above. Too few colonies to count would be 30 or below. So technically speaking, we have 1 plate here that is countable. So you did all that work, spent all that time, did all of that stuff, and you get 1 plate on which you count 159 colonies. And that doesn't mean that at that point in time there were 159 colonies per milliliter in the original sample. It means that there were 159 colonies in that dilution. So we have to fix the math so that the number that we're using is not what was in the dilution, but was actually in the sample. And that's the beauty of scientific notation. All you have to do is take away the minus sign. And instead of being a 10 to the minus 5, we now have 159 colony forming units times 10 to the minus 5, or excuse me, 10 to the 5. 159 times 10 to the 5 colony forming units per mil in that original culture. So in that culture, at that period of time, there were 159 times 10 to the 5 cells per mil of culture, 15,900,000 cells per mil. So you can imagine you would never have been able to count that, but you can count a dilution of that. So the benefit of the serial dilution and spread plating protocol, and what makes it worthwhile, even though it's a time hog, it's a media hog, it takes you a lot of effort, is that you get a viable count at the end of this. You don't get a count of all of the cells that are there. You get a count of the cells that are capable of undergoing binary fission, and those cells we call viable. And we signify the fact that this is a count based on a viable method by not saying 1.59 times 10 to the 7 or 159 times 10 to the 5 cells per mil. We say colony forming units per mil, or CFUs per mil, in order to say to anyone who reads those data, that was a viable method that they used. They don't even have to look at your protocol. They know by the fact that you used CFUs that you did a viable method of counting those cells. And the thing that makes this direct is because you were looking at colonies, directly at the bacterial number. Another direct method that you can use to count microorganisms is a microscopic count. Now this one, because you're just looking at the cells under the microscope, is not a viable count because both live and dead cells would be visible on microscope slides. So this is considered a total method. It's still considered a direct method because what you're counting is actually the cells. So one of the drawbacks for this is that you have to have this special kind of microscope slide, which has a cover slip attached to it and has this quantified area into which a known quantity of the bacterial sample will go. And that allows you to just count what you see under the microscope and apply a formula that's given to you by the company who sells the microscope slide, and then you get a number of how many cells per mil. So you add the bacterial suspension underneath the fixed cover slip, and then you put that under the microscope and you count one of the cells. So in this example, you have 14 bacterial cells in that larger square, and you will apply to that the formula that this square is 1,250,000th of a milliliter, which means that you have to take your number and multiply it by 1,250,000. And so at this point in time, you have 17,500,000 cells in a milliliter, which you could apply scientific notation to. And that would give you 1.75 times 10 to the 7. Notice here, though, cells per mil, not colony forming units. And so when you read this, you know that these data were reflective of the total cell count and not the viable cell count. So that's one of the drawbacks with this direct method. The other is that you have to buy one of these microscope slides. You also have to manage to wrangle the bacteria enough to count them, which could be difficult, especially if you have motile bacteria. The benefit of this one, though, is that you can get a very quick count. There's no incubation time. It's relatively inexpensive supply-wise because all you need is this microscope slide. So it's fast, but it can be fairly challenging to count these bacteria. So notice we now have one direct viable method of counting in which we record CFUs per mil. We have one direct total method of counting. And now we're going to move on to our indirect methods, which we will have two examples of as well. The first of those is the indirect viable method, which is called the most probable number test. And in the most probable number test, instead of diluting in a series, you dilute straight from your culture. So this is something that you wouldn't be concerned about taking large amounts from because you've taken a sample that you're not going to do anything with. You're going to destroy that sample. So for instance, in the first set, you would inoculate 10 mils of whatever that environmental sample was into each of those 5 tubes. And in set 2, you would inoculate 1 mil. So notice that's a 1 to 10 dilution, or a 10 to the minus 1 dilution. And then in set 3, you would put 0.1 mil. And now you would have something that was 10 to the minus 2, or 1/100th, as concentrated as what was in that first sample. Then you take them and you put them into the incubator and you allow them to grow, which means that this is a viable count. You look for growth, and growth tells you whether or not you have organisms present. So this is a viable method. The reason that this method is indirect is because this method does not look at cells or colony directly. This method looks for turbidity in the broth as evidence of growth. So if there is turbidity, the increase in the turbidity is assumed to be because microorganisms are present and growing. So after you incubate these, if in the first set, the undiluted set, you get 5 positive, so 5 of those tubes are turbid, and then in the second, 10 to the minus 1 set, 3 of the tubes are turbid, and then in the third 10 to the minus 2 set, one tube is turbid. You know that there's something there. But the cool thing about the MPN is it will give you a most probable number for the amount of organisms that are actually in that particular sample. Thankfully, it involves no math. It's very easy. You get this code 531, that's your MPN code, and you apply it to an MPN table, which you can Google and download. And when we take that 531 code and look at the MPN table, notice the MPN code 531 gives us a most probable number of organisms per 100 mil of 110. You might think, wow, that's pretty slick. I get a number, 110 sounds good. The issue with the most probable number, though it's pretty easy and though it gives you a viable count, is that in order to be very confident of that number, you have to give a very wide range, right? So statistics are really helpful in helping us to understand just how much we can trust numbers. So the MPN has something called a 95% confidence limit that's reported along with the most probable number. And notice for the code 531, the most probable number is 110. But if you want to be 95% sure that you actually have the right number, you have to say it's somewhere between 34 and 250 cells. So this will tell you the viable number of cells. So we would report that as 110 colony forming units per 100 mil. But we'd also have to put with it the caveat that we're really not that confident in that number. If we really want to be confident, if somebody says, I want you to be 95% sure of the number, you have to say, well, it's somewhere between 34 and 250. So it doesn't give us a really confident number, but it gives us a pretty easy number. It's relatively quick. And so typically what most probable number tests are done to do is just a screening. Because if this is your food, you don't want there to be 110 or 34 or 250 microorganisms in your food. So this will be a screening test. And then further testing will be done that is more precise in its number. But the MPN will give you a number that you can start working with, and you can rule out the possibility of microbial contamination or rule it in and then work on it further. So that's our indirect viable method. Indirect because we're looking at turbidity and assuming it's equivalent to an increase in cells. Viable because it requires growth. And so we know that dead cells are not being counted. And further than just saying, oh, one's turbid and one's not turbid, which would be a qualitative assessment, we now have a quantitative assessment where we have a number. So notice for each of these, whether we're talking about serial dilution and spread plating or microscopic counts or most probable number, or the next thing we're going to look at, which is spectrophotometry, you're getting a number, which makes each of these quantitative assessments, right? Quantitative assessments give you numbers. Qualitative assessments give you a general idea, like turbid or not turbid. And if you want to think about that and maybe something that's a little more personally applicable, think about the difference between someone telling you it's hot outside and someone telling you they looked at a thermometer and it's 95 degrees outside, right? Hot outside is relative. The number is something that exists outside of the opinion of the person measuring it, right? So if somebody tells you 85 degrees, you know what 85 degrees feels like to you. And each of these methods that we're talking about give us a number, and so that's very helpful to us. Next up, the indirect total method, spectrophotometry, which means to measure using the light spectrum, right? Metry means measure, spectro means the light spectrum, and photo, of course, means light. We know that from photosynthesis. So what this is a really cool machine that takes the word turbidity and applies a number to it, and it does that by shining a light through your culture and determining how much of the light is being absorbed by the culture, which is exactly what you do when you look at a turbid tube. You don't think about it in those terms, but when you say something is turbid, what you're saying is I can't see through it. So this machine takes that phrase, I can't see through it, and it applies a number to it. So at the top here, we have a picture of a light shining onto a culture that has no organisms in it. So notice it has an absorbance of 0 or a transmittance of 100%. But when we have lots and lots of cells, they absorb the light, which keeps the light from being transmitted through, and you end up with an absorbance reading of 0.7 or a transmittance reading of 20%. Now, typically, we read the absorbance because as the culture grows, the absorbance goes up, and that allows us to plot things on a positive axis. We utilize this method of counting cells in order to determine the pH and the temperature optima of our organisms in lab exercise 11. Because the differences between those can be very subtle, we don't just want to say, oh, this is turbid and this is not as turbid. We want numbers. We want a quantitative assessment. So we use that spectrophotometer to give us some quantitative data. And when we get those quantitative data, then we can make comparisons that exist even outside of the data set that we're working with at a particular moment in time. Now, just to kind of give you an idea, since you may not have looked at those lab data yet, these are some data that were taken from the test tubes that you see at the bottom of your screen. So in those test tubes, we have a test being done to determine the pH optimum of this organism. So it's grown at pH 6 on the very left-hand side, pH 7, pH 8, pH 10, and pH 11. And then next to that, there's a positive control, which is grown at the natural pH of the environment where it was found, and a negative control, which is just that medium with no cells inoculated in it, right? Because we need to know how turbid just the medium is. So you can see that positive control, that has a ton of growth of this purple sulfur bacterium. It's really, really dark purple. And so we set that as maximal. And then we compare each of the experimental broths to that maximum control. But the cool thing about this is it takes the turbidity we see in the tubes and gives us a number, right? Notice the y-axis is the absorbance relative to the control. The last method that we're going to talk about is something called flow cytometry, which is not used a lot in conventional microbiology, but it is something that we're going to be talking about later on. So I want to introduce it here so at least you have some context for it later on. And a flow cytometer is a cell counter, right? Cyto means cell, metry means count. So this is a cell counter that counts cells as they flow through a machine. And like I mentioned earlier, this is one that I think makes a lot more sense if you watch it happen dynamically. So I'm going to show you a quick video in order that you see this process happening and understand how these flow cytometers can count microorganisms. Now, this is an indirect method, and it is a total cell count method. Typically, it counts live and dead cells, though you can tweak the flow cytometry a little bit and make it only count viable cells. Typically, this is an indirect total method of counting.
In this animation, we will introduce you to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell's size, shape, and complexity, a flow cytometer can also detect emitted light from excited fluorescent molecules, such as fluorescently labeled antibodies or fluorescent dyes or stains. For the rest of this animation, we will focus on the detection of fluorescent signal by the flow cytometer. For more information about fluorescence, please see the fluorescence animation. The fluorescent molecules, or fluorophores, are excited by the correct wavelength of laser light when the cell passes through the laser beam. After excitation, the light emitted by the fluorophore is directed along a path with emission filters that allow for the detection of multiple different fluorophores emitting light in a cell. A flow cytometer can detect the presence of a fluorophore and also quantify the relative amount of a fluorophore within a cell. A flow cytometer can also be used to detect light emitted from multiple fluorophores in the same sample. While it is common for flow cytometers to detect 2, 3, or 4 colors at the same time, some flow cytometers can even detect up to 18 different colors at the same time. The data can then be presented to the researcher in the form of a histogram or a dot plot. Such data furthers the powers of flow cytometry analysis by enabling the researcher to analyze individual populations of cells within a heterogeneous mix of cells in the sample.
One last thing before we wrap up the counting of microbial cultures and move into microbial growth requirements is to talk about something called a biofilm, which is a really cool thing. Well, it depends on where it's growing. The electron micrograph at the bottom left-hand side of your screen is a biofilm that's growing on a catheter, and it has Staphylococcus aureus embedded in it, and that means that this person has a very serious infection. So that's not a cool thing from the human perspective, but from the perspective of the microorganism, the ability to produce something called a biofilm is a really cool thing to be able to do because it allows the cells to exist in this highly protected environment where they can share nutrients and remove waste and even plan and coordinate reproduction. And from our perspective, it's very bad because it allows those microorganisms to resist antibiotics and all sorts of things that we're going to talk about when we talk about trying to control microbial growth. So a biofilm happens when microbial cells land on some surface, and that could be the rim of a bucket, it could be a urinary catheter, it could be a prosthetic joint, and that attachment, because they produce chemicals, happens very, very quickly, and then they become irreversibly attached to that surface fairly quickly, and then they undergo binary fission and grow and grow and grow and grow, and then they produce this EPS and form water channels. So they're almost like this city of microorganisms, and they grow across the surface of things, and then as they grow, they can actually release new microorganisms to disperse off into new sites in the patient or on the bucket that's holding the water sample, or in your drains of your sinks, or in the ducting of your air conditioning. And they're really, really nasty little things in terms of human infection, because they make it very, very difficult for us to remove microorganisms and treat bacterial infections. And like I said with the last slide, the biofilm, again, is one of those things that I think is helpful to see happen dynamically. So I'm going to add a little video here so you can see that.
The life cycle begins when bacteria enter the bloodstream from various infection routes and evade the immune system or antibiotics. Formation begins with attachment of planktonic bacteria to the surface, preconditioned with proteins from the host immune system. Irreversible attachment to host proteins signals the bacteria to create biofilm and is the first committed step of biofilm growth. The bacteria anchor themselves permanently using cell adhesion structures such as pili and receptors called epitopes. This encourages new bacteria to attach to newly arriving cells, providing adhesion sites and building the protective matrix of the biofilm structure. Cells now communicate via quorum sensing and use small molecules to regulate gene expression within the bacterium. The community grows through both cell division and recruitment. The development of this complex biofilm makes the community resistant to the immune response and antibiotics. It may include diverse species of bacteria, yeasts, minerals, and host proteins. Recruited neutrophils result in a cascade of inflammatory cytokines and host proteases. These damage tissue and regulate fluid accumulation and exudate, providing nutrients. Bacteria now exist in multiple forms and metabolic states. Now a different phenotype, both physically and metabolically, it has the survival advantages of diversity and multiple defenses. The life cycle continues as fragments or planktonic bacteria are released to reinfect the human host.
All right, the second part of this lecture concerns itself with the requirements that microbes have in order to grow well. So we're going to talk about growth requirements that are physical in nature, and then we'll talk about growth requirements that are chemical in nature. And all of those things are meant to determine for us the optimal environments in which these microorganisms grow so that they reach their biotic potential. So what physical and chemical things can we change or manipulate in order to get these microorganisms to grow optimally? First up, we're going to talk about life in the presence of oxygen, which is kind of interesting to find out is actually fairly difficult to accomplish, because oxygen will naturally convert to what are called reactive oxygen species. So reactive oxygen species, which you'll sometimes hear called toxic oxygen species or free radicals, are chemically reactive oxygen species. So molecular oxygen, O2, is very stable, but the problem is that oxygen molecule, as it's being manipulated in the cell, can be converted to these reactive oxygen species or free radicals, and they're incredibly damaging to the cell. They convert to superoxides or hydrogen peroxides, and those things happen as a result of aerobic respiration. And once those superoxides and hydrogen peroxides are produced, they're kind of, if you picture like the Tasmanian devil, they just spin up and they zoom all over the cytoplasm of the cell, and as they bang into DNA or RNA or fatty acids or amino acids or enzymes or anything else in the cell, they just bang into them and break them. And if they break enough of them or cause enough damage to the DNA, it kills the cell. So if you are going to live in the presence of oxygen, you have to have enzymes or some ability to deal with these reactive oxygen species. You might remember me mentioning reactive oxygen species, when we were talking about microbial cells, I mentioned that there are magnetosomes in which cells use iron oxide to destroy hydrogen peroxide. So that's one option that you have if you're living in an aerobic environment. Typically, we think of two enzymes being used in order for cells to be able to grow in the presence of oxygen, and those are superoxide dismutase and catalase. And these are considered antioxidants because they undo the oxidation of that reactive oxygen species and convert it back to its reduced non-reactive form. So if you take this superoxide anion and you expose it to a superoxide dismutase, the superoxide dismutase will convert that superoxide anion to hydrogen peroxide. So now you don't have that superoxide anymore. The problem is you have hydrogen peroxide. So most of the time, superoxide dismutase is found in organisms which also have a second enzyme, catalase, which converts hydrogen peroxide to the very stable and non-reactive water molecule. So calling this an antioxidant might help you to connect it to the real world, right? People talk about free radicals and talk about taking foods that are rich in antioxidants in order to help them to deal with free radicals that can contribute to disease or even aging, right? So wrinkles are thought to be a result of free radical damage to skin cells. So if you take an antioxidant lotion or topical ointment, you can assumably undo some of that damage. But what we're talking about is the damage that causes death to cells, not single cells of a multicellular organism, but the cell of a unicellular organism, which causes it to die. One of these two, superoxide dismutase, we don't test for in lab, but we do test for catalase because it's a really easy test to do. All you have to do is take a colony of cells and add hydrogen peroxide to that colony, and if the colony bubbles, you know that it produces catalase. If the colony does not bubble, the colony is a catalase negative organism. So in this photograph, you have on the left-hand side a catalase positive organism, and on the right-hand side a catalase negative organism. In the next slide, we're going to talk about how the presence of catalase and superoxide dismutase allow organisms to live in and thrive in aerobic environments. And we've talked a little bit about oxygen in the past. You've seen some stuff in lab about the growth requirements of organisms in terms of their oxygen requirements. And you know that one of the media that we use in order to determine the oxygen requirements for a given microorganism is something called fluid thioglycolate media. And fluid thioglycolate media is described in detail in lab exercise 11. What I'd like to do here is not so much describe the medium, but describe what happens and what that can tell us about microorganisms. So FTM has a reducing agent in it, which takes the oxygen that diffuses into the medium from the environment and reduces it to water, creating an anaerobic environment throughout about the bottom, say, 70 to 80% of the tube. So when we inoculate bacteria into this medium, they will attach themselves to tiny pieces of agar and they won't move vertically through the media. So where you put them, they will either grow and make the medium turbid there, or they will die because whatever oxygen level is there is not appropriate for them. And so where they grow in this media can tell you what their oxygen requirement is. So in group A, you have organisms who will only grow at atmospheric concentrations of oxygen, and we call those organisms obligate aerobes or strict aerobes. Strict aerobes, like you and I and like other organisms like Bacillus subtilis, will only grow in the presence of oxygen. However, they have to have special enzymes in order to prevent the reactive oxygen species that are produced as a byproduct of aerobic metabolism to protect themselves. So they produce both catalase and superoxide dismutase to protect themselves from the reactive oxygen species that are being created as they undergo aerobic respiration. The second group are the exact opposite of that. Those are obligate or strict anaerobes, and those guys will only grow in environments where oxygen is not present. And the reason for that is that these organisms lack those enzymes that would neutralize the reactive oxygen species. And so when oxygen is present, the oxygen converts to these reactive oxygen species, and then the organism dies in the presence of oxygen. So they're only capable of growing where there is no oxygen present. Next, we have two really interesting organisms. Notice both of their names have anaerobe in them. We have a facultative anaerobe and we have an aerotolerant anaerobe. So both of these organisms can grow anaerobically, as is evidenced by their growth in FTM. They also can grow aerobically. So they are not strict or obligated to anything. They can grow in both environments. The difference between the two of them can be seen in the word that precedes anaerobe. The facultative anaerobe is an organism that can grow anaerobically, but does not prefer growing anaerobically. Whenever you see facultative, whatever follows it is not its optimum. It's just something it's capable of. So a facultative anaerobe can grow without oxygen, but it does not prefer that. So their growth pattern is to grow really well in the presence of oxygen and to grow not as well in the anaerobic environment. And that has nothing to do with these neutralizing enzymes, superoxide dismutase and catalase. It has more to do with the fact that when oxygen is present, they will undergo aerobic respiration, which, as you know from previous lectures, gives you the most ATP per pound. And so they can make way more ATP, they can grow way faster when oxygen is present. When oxygen is not present, they will either switch to fermentative metabolism and make very, very few ATP per molecule of glucose, or they will switch to anaerobic respiration, which, though it is better than fermentation, still does not produce the amount of ATP that it's produced during aerobic respiration. So they will grow anaerobically. They do not prefer it. They are facultative anaerobes. Next up, we have our other anaerobes that have a caveat to their name. These are the aerotolerant anaerobes. So that tells you what they're doing. Unlike the obligate or strict anaerobe, these guys can tolerate an aerobic environment. They tolerate oxygen, but they never use oxygen. So oxygen has no effect on their growth. It does not increase their growth like the facultative anaerobe, and it does not inhibit their growth like the obligate anaerobe. The aerotolerant anaerobe can grow in the presence of oxygen, but it does not utilize the oxygen, so it doesn't increase its growth. And these guys have superoxide dismutase to neutralize the reactive oxygen species, but they either are fermentative, like genus Streptococcus, or they just don't grow preferentially with oxygen because they lack catalase. Finally, we have the microaerophiles. And microaerophiles, they like oxygen, but they like just a little bit. So these guys grow in 5 to 15% oxygen. And if you increase the oxygen level above that, then the reactive oxygen species that are made will become lethal and the organisms die. So they cannot grow anaerobically and they can't grow in full atmospheric oxygen. So when we grow bacteria in FTM, it's super cool, right? Because in this one tube, we can tell not only can you grow aerobically or anaerobically, but do you use oxygen or do you just tolerate its presence? One other way that we can look for the ability to grow anaerobically is to use an anaerobic jar, which is pictured here on the left, which, like the fluid thioglycolate medium, has a reducing agent which converts all of the oxygen in that sealed container to water. And if you're working with a strict anaerobe, you can do your inoculations in an anaerobic hood. So rather than doing those inoculations on the lab bench, which would be exposed to oxygen, you can actually perform your inoculations in one of these anaerobic hoods. So one example, I know this is kind of disgusting, but one example of an anaerobic microorganism that's clinically important is that same Clostridium perfringens that I mentioned has a 6-minute generation time, because that is capable of growing in anaerobic tissues and can very quickly cause gas gangrene, and that's a very common issue for people who have diabetes. They have poor circulation into those tissues. The anaerobic tissues, because they have poor circulation, don't have a lot of immune system interaction. They also tend to have neuropathy, so they can't feel their feet. They don't know that they have that infection, and that infection can very, very quickly get out of control because of that very rapidly reproducing anaerobic microorganism. Next, we have pH. So organisms all have a fairly narrow pH zone in which they do well. And on either side of that very narrow zone, they very quickly don't grow at all. So if we take the middle of the pH scale, the pH 7, we have a neutral pH, and organisms who thrive at a neutral pH are called neutrophiles. Organisms who thrive at an acidic pH are called acidophiles, and organisms who thrive at a basic or alkaline pH are called alkalinophiles. Notice again on both sides, right, whether it's too acidic or too alkaline, they very quickly perish and are not able to grow at all. Now, regardless of the environment you're talking about, when the pH gets too low for the acidophile or the neutrophile or the alkalinophile, that causes the organism to tumble if they're motile. So they will all try to move out of that low pH environment and hopefully move into one that is more at their optimum. In addition, in all of these environments, the surface proteins of these microorganisms are incredibly stable, and they adapt very quickly to changes in the pH. In addition, the proteins that are found on the membrane or surface of the cells will create these really interesting microenvironments where they actually gather protons in order to neutralize an alkaline environment, or they grab hydroxyl ions from the environment in order to neutralize an acidic environment. They also buffer changes in pH using their nucleic acids, using the phosphate backbone of their nucleic acid. They buffer their environment, and they have basic or alkaline or acidic amino acid side chains, which allow them to adapt to environments. So in any environment, whether you're an acidophile, a neutrophile, or an alkalinophile, low pH makes you move out of the environment. Your surface is very stable to changes in pH and can change, right? It can change and gather protons to offset an alkaline environment or gather hydroxyls to offset an acidic environment. If they get inside the cell, the DNA and the RNA can serve to buffer those so it can lose protons in order to neutralize and bring down the pH, or it can gather protons in order to neutralize and bring up the pH, and then their amino acid side chains can gather protons and hydroxyls as well. And it's really crazy. So whereas our cells have a very narrow pH within which they can operate, right? Typically we think of the pH for human cells to be 7.4 or thereabouts, right? 7.35 to 7.45 or something like that. E. coli, which is a neutrophile, you can put that cell in a pH range from pH 4.5 to 7.9. So let's just say 5 to 8. And the inside of that E. coli cell, the cytoplasm, will only register a 0.1 unit change in pH. So you have an environment, right? Remember pH scale, each number is a tenfold difference. So when you say from 5 to 8, 5 to 6, so 6 is 10 times more alkaline than 5, 7 is 100 times more alkaline than 5, 8 is 1,000 times more alkaline than 5. pH 8 is 1,000 times more alkaline than pH 5, and the inside of that cell will only change 0.1 units. So they're incredibly adaptable and able to withstand changes in pH, much more so than human cells or animal cells. And that allows them to inhabit these environments that change very, very quickly and routinely and adapt to those changes. Now, when you talk about cells who live in an alkaline environment, those that are alkalinophiles, they typically also live in salt-rich environments, and they utilize sodium and proton antiporters. So they utilize this active transport, much like the sodium-potassium pump you talk about in general biology, in order to move sodium along its concentration gradient and then pump protons opposite their concentration gradient. So they have these voltage-gated channels where sodium moves in and then sodium is pushed out as hydrogen ions are brought in. So that's how they work in alkaline environments. In acidic environments, those cells who are acidophilic tend to have membranes that are not very permeable to protons at all, but they also will utilize that natural proton gradient in order to undergo chemiosmosis. So they don't even have to use the electron transport chain. All they have to do is allow those protons to flow down their proton gradient from the outside inside, and then they just collect up those protons and expel them in vesicles in order to get the protons back out of the cell. So all cells are capable of living in a much, much wider range of pH than animal cells or fungal cells or plant cells. And that's because they can move out of that low pH environment. They have very pH stable proteins and amino acids. Their membranes will create a microenvironment around the cell that is more neutral than the surrounding environment. And if the protons or hydroxyl ions get into the cell, the DNA chromosome, the phosphate backbone of the chromosome, can be used to buffer and keep the internal pH very stable, even though the external pH might vary wildly. So one example of acidophilic microorganisms is one who also is a thermophilic organism. So in Yellowstone Park, in the geysers, we have these huge pools where thermoacidophilic microorganisms both survive, thrive, and require hot environments and acidic environments. In addition, there are haloalkalophilic microorganisms that like to grow in both salt-rich and alkaline environments. Next up, we have temperature. And the reason that temperature is important to microorganisms is that temperature has an effect on enzymes. And that's something that we talked about in the metabolism lecture a little bit. At optimal temperature, enzymatic reactions are occurring at a maximal rate. Anything above that optimum is in danger of denaturing the proteins and perhaps lysing the cell due to heat. Anything below that slows the interaction between substrate and enzyme, and because substrates and enzymes are not encountering one another, ATP is not being produced and the cell is growing very slowly. Now, one thing to note that's very important, notice I said at higher than optimal temperatures, the cell dies. Proteins denature, they don't come back after they denature, and that will kill the cell if the cell gets hot enough that it undergoes thermal lysis. Obviously, the cell doesn't come back from being blown up. So high temperature, higher than optimal temperature, kills microorganisms. Now notice on the low end, minimum growth does not become minimum because of death. It becomes a minimal temperature at which the organism will grow, because at that point, the membrane becomes so solidified that nutrients can't be transported into the cell, and enzyme reactions are going so slowly that growth does not occur. So minimum temperature does not kill the cell. So low temperature can control the growth of a cell by slowing growth, but it does not kill cells, whereas maximal temperature exists where the cell actually dies. So notice there are no degrees on the x-axis. It's simply temperature, and that is because the optimum for organisms varies widely when we talk about microorganisms. Those organisms who grow optimally at body temperature, right in the middle of this range, are called mesophiles, right? They're the middle likers, right? Middle, meso means middle. So a mesophile likes it in the middle. They like it right at body temperature. So at higher than body temperature, enzymes start to denature. At lower than body temperature, everything's going slower, slower, slower. Those organisms who grow, thrive, and inhabit higher than body temperature environments are called thermophiles. And thermophiles, typically, we talk about growing optimally at 60 degrees Celsius or higher. Hyperthermophiles grow optimally at or above the boiling temperature of water. And then on the low end of the scale, psychrophiles grow at between 4 and 10 degrees Celsius optimally. And psychrotrophs are a really interesting group of organisms. So psychotroph, you could kind of compare to the facultative anaerobe, right? Whereas the facultative anaerobe can survive anaerobically but doesn't prefer it. The psychotroph can grow in cold, but it doesn't prefer it. So psychrotrophs are those organisms which can grow in cool temperatures but don't prefer it. And really, for this one, the psychotroph, the problem with a psychotroph is his optimal temperature for growth is usually higher than what is indicated on this chart here from the Tortora textbook. Psychotrophs typically grow optimally at mesophilic temperatures, but can grow at low temperatures. And the problem for us with psychotrophs from a human perspective is that they can cause food spoilage because they're able to grow relatively well at refrigerator temperatures. So a mesophile doesn't really grow at refrigerator temperatures, whereas a psychotroph can grow at refrigerator temperatures and cause food spoilage. And just to talk about thermophiles, so thermophiles a lot of times are also barophiles. They require high pressure, and a lot of them exist in the deep sea hydrothermal vents. This is a barothermophilic community in the North Pacific Ocean off Vancouver Island in the Juan de Fuca Ridge. So again, kind of interesting, we have these polyextremophiles. We had haloalkylophilic organisms, thermoacidophilic, and now we have a barothermophilic microorganisms existing at high temperature and high pressure. All right, the last thing that we're going to look at for physical growth requirements is salt. And the reason we look at salt is because salt can cause osmotic or trigger pressure based on the relative concentration of salt in the environment and in the cell. When the concentration of salt outside is lower than inside, then water wants to rush into the cell, and that can cause the cell to lyse, right? Which is one of the reasons why bacteria like to have cell walls, because cell walls resist that Turgor pressure, right? So I made Turgor big there, so you think, oh, Turgor pressure causes the cell to get big. Now, if the concentration of salts and solutes inside the cell is lower than the outside of the cell, then that can cause things to leave, and the cell will shrink under what's called osmotic pressure. So osmotic pressure causes the cell to shrink. Turgor pressure would cause the cell to get larger. And the real problem for most organisms is that they encounter osmotic stress they live in an environment where they have a high concentration of things inside their cells and water wants to rush in, right? So how do they deal with that? They have a cell wall which resists that turgor pressure. But you can live in an environment where you have more things outside, like a high salt environment like the ocean or the Dead Sea or the Great Salt Lake or Mono Lake or something like that. And in that environment, you exist in a state where water constantly wants to leave the cell, right? Osmotic pressure makes the water want to go outside and dilute what's out there. And so you have to come up with a way of keeping the water inside of the cell. So basically you have to come up with a way of having enough stuff inside that the water is balanced. So in order to do that, cells will make what are called compatible solutes. And compatible solutes increase the halotolerance of the cell. And they have to have a couple of traits in order to make them work. Number one, they have to have a high solubility in water. So you have to be able to have a lot of these compatible solutes inside the cell that can be dissolved in water. They also need to have a high permeability so that if the osmotic stress changes, they can go outside of the cell. They also, very importantly, unlike salt, need to be metabolically inert so that they don't damage any of the portions of the cell. So they increase the halotolerance of the cell by making water want to stay inside the cell, and they have to have a high solubility, high permeability, and they need to be metabolically inert. And there are several different kinds of compatible solutes. So we don't typically talk about low salt environments, we talk about high salt environments. And we compare those to the salt that our cells like. So if we are non-halophilic, we consider non-halophiles to like a salt concentration of 0.09%, and anything that likes it higher than that, we give a halo prefix to. So halophiles like it to be saltier than 0.9, and typically they want it to be at least 3% salt. So that's marine organisms. Marine organisms, the ocean is 3% salt. Halotolerant organisms do not prefer a high salt environment, but like the aerotolerant anaerobe can tolerate a high salt environment. So they grow best in a non-halophilic environment, but they can grow in fairly high concentrations of salt. Extreme halophiles grow, thrive, require concentrations of salt that get toward the saturation point of salt in water, right? So they can grow at 30% salt, and it doesn't get any higher than that. If the salt concentration gets any higher than 30%, the salt comes out of solution as crystals. So here's one other example of haloalkalophilic microorganisms that's shown in the OpenStax textbook, Lake Natron in Tanzania. And that red color that you see is actually a pigment produced by those haloalkalophilic microorganisms, and that pigment actually helps them to survive in the presence of high salt. And now we're going to switch our attention to chemical growth requirements. Cells typically have 6 chemical growth requirements. They require carbon, they require nitrogen, they require sulfur, and they require phosphorus. Carbon, of course, is the backbone of all of the biological macromolecules, proteins, polysaccharides, nucleic acids, and lipids. Nitrogen is found in nucleic acids and proteins. Sulfur is found in proteins. Phosphorus is found in nucleic acids. And so all cells require some form of carbon, nitrogen, sulfur, and phosphorus. Cells, just like your cells, also require vitamins and minerals. Trace elements are the vitamins that are required by a particular microorganism, and organic growth factors are the minerals that are required by these organisms. So as you can see here, they require proteins both for their flagella, their membrane proteins, their channel proteins, pumps, and all that sort of stuff, receptor proteins. The cell wall has a protein component. There are proteins in the cytoplasm. Nucleic acids, of course, the DNA is found as part of the chromosome and the plasmids. RNA is found in the ribosomes, the messenger RNA and the tRNA. Polysaccharides are found in the glycan portion of the cell wall. They're found in the lipopolysaccharide outer membrane. And of course, polysaccharides are storage granules are filled with polysaccharides as an energy source. Lipids are part of the lipopolysaccharide outer membrane. They're part of the phospholipid bilayer membrane. And of course, fats are used as a storage molecule as well. So you have polysaccharide granules and lipid inclusions and all that fun stuff in order to store energy for long-term use. So when we're growing microorganisms, we work really hard to figure out what kind of carbon do they like, what kind of nitrogen do they like, what kind of sulfur, what kind of phosphorus, what vitamins do they need, what minerals do they need in order to grow well, because most of the time in the microbiology lab, we're trying to get our microorganisms to grow well so that we can learn about them. So I want to take just a moment to show you a couple of experiments and how very small changes in the chemicals that we provide to microorganisms can cause very drastic changes in their rate of growth. So first off, we're looking at a microorganism who's grown in a medium whose nitrogen source is inorganic. The nitrogen source that's being fed to this organism is ammonium chloride. And when we do that, the generation time of this organism, not 6 minutes or 20 minutes, but 85 hours, which means that you have to wait a very long time to learn anything about this organism. So you start tweaking the recipe and trying to decide whether or not you can figure out a better way of feeding this microorganism. And one of the things that you can change is the ammonia. You can say, okay, instead of giving him an inorganic form of nitrogen, I'm going to try an organic form of nitrogen. So you give him glutamine, which is an amino acid, and lo and behold, the generation time drops from 85 to 30 hours. Now, that's still not 6 minutes or 20 minutes, but it's about a third of what the generation time was previously. So sometimes making minute changes in the media makes for drastic changes in the generation time and the biotic potential of the organism that you're growing. Now, for us, that might mean differences between these two media here. For instance, on the left, you have nutrient agar, which is a complex medium for the growth of heterotrophic bacteria. So it's complex, which we talked about earlier on in lab, which means we don't really know exactly what's in it, but it works pretty well for most heterotrophs. It's got peptone in it, it's got beef extract, and it's got some salt. Now, if we want to be able to grow Escherichia coli maximally, we might switch from nutrient agar, just this complex medium, to something that's been chemically defined, where now we know exactly how much of every single thing is inside this medium. And when we do that, we can grow E. coli faster. The question you want to ask yourself is, does it really matter whether the generation time is 20 minutes or 30 minutes? Is that really going to change things? And is that change great enough that I want to have a chemically defined medium for every single microorganism that I want to grow in my lab? And for the most part, in a general lab, no. The answer is no. You don't need to get the generation time down to 20. 30 is just fine, because 30 minutes will still provide you with ample growth of the microorganism in a 24-hour period. So most of the time, we don't work with chemically defined media for microorganisms. We work with complex media, because complex media like nutrient agar or Luria bertani agar or tryptic soy agar or things like that will grow heterotrophs very nearly optimally, and that's good enough for us. When we're talking about something like the isolate I just showed you data for that can go from 85 hours to 30 hours, now you're talking about a change that's significant. And I want to show you, not because I want you to memorize any of these details, but I want to show you just how small a change that really is. This medium here is all of the things that go into a medium for growing purple sulfur bacteria photoautotrophically. One of those ingredients is nitrogen. They need a nitrogen source. In the first example, which yielded an 85-hour generation time, we used 0.5 grams per liter of ammonium chloride, which we switched to glutamine for the second experiment and reduced the generation time to a third. So even small media changes really do matter. And if you're talking about something that has a long generation time and does not have very high biotic potential, then this kind of a change might be something that you want to investigate. But for us, we normally just grow hemoheterotrophs in a general complex medium like nutrient broth or tryptic soy broth or something like that.